Towards the end of the 19th century, a great deal of progress was made in uncovering the nature of the nervous system. The nervous system had been something of a puzzle. It had become clear over the previous 150 years that bodies were composed of tissues and tissues were composed of cells. Now, there are many kinds of tissues in the body, and the tissue of the nervous system posed particular difficulties. It was very, very difficult to resolve through a microscope the individual cells. And it was the last tissue that ultimately gave up its secrets and was shown to be composed of specific kinds of cells. The lion's share of attention here will be given to those cells called neurons. They are not the only cells in the nervous system, and they are not necessarily the only efficacious cells in the nervous system. But neurons have hogged the limelight, and at the end of the 19th century, a physiologist called Camillo Golgi invented a staining technique using silver nitrate that allowed, for the first time, the structure of individual cells to be seen in all their complexity. So he published this in 1873, and the pictures that resulted were quite stunning. Now, photography was itself a very young art at this stage, and despite the fact that the staining technique meant that details of individual nerve cells could be seen clearly through the microscope, it turned out it was better to draw what you saw through the microscope than to attempt to photograph it. And so what we have from this time then are some of the original drawings illustrating the structure of various elements within the nervous system, both peripheral and central. Here, for example, is a drawing by Golgi of the hippocampus. You can see an awful lot of detail. You can see individual cell bodies, and you can see the long filaments coming out of them. This is a peculiarity of neurons. They have very, very long associated filaments. Here's another drawing by Golgi of a very select few cells from the cerebellar cortex. The cerebellum is a part of the brain at the back and low and phylogenetically of older origin than the neocortex, which we often think of when we think of brains. So Golgi had made a huge step forward here. But there was another physiologist, this Spanish guy, Santiago Ramón y Cajal, who adopted Golgi's technique, and to everyone's chagrin, he turned out to be better at it than Golgi. He was a better draft person, his drawings were clearer, and his staining was superior. He managed to resolve more detail than did Golgi. So here's some of Cajal's drawings. They are absolute stunning works of art. There are very many of them available. These show two particular kinds of cells, the Purkinje and the granule cells from the pigeon cerebellum, and you can see the fine, fine detail that is resolved in these images. Here's another one from chick cerebellum from 1905. These are fantastic landscapes that nobody had ever seen before. And importantly, Ramón y Cajal managed to make clear that there were gaps between the ends of one neural filament and the neighboring adjacent neuron. These we call these synaptic gaps, and they underlie a very important debate that was had at the time, which we're about to dive into. You need to know something about the basic structure of a neuron. Here, what's shown here are 3 neurons standing in for the entire nervous system. The sensory neuron has receptor endings, which you might imagine as being coming from the retina or the inner ear or the skin or the mucous membranes of the nose or mouth. And they are stimulated by something which causes electrical activity, which goes up when, in a manner that we'll see, it travels in discrete packets called spikes, past the cell body and down to a bunch of branches called dendrites. And from there, the electrical signal ultimately passes to the next neuron, but as we'll see, through a chemical mediation. Now, there's a single interneuron here, and you should consider this as standing in for all brain neurons. For brain neurons do not connect to receptors directly, and they do not connect to muscles directly. Interneurons are neurons which receive electrochemical signals from other neurons and pass them on to other neurons. So all they ever know is neurons, is this electrochemical, purely internal business. But there are some neurons which project then away from the brain. And the red neuron on the bottom here shows this. This is a motor neuron which terminates not in connections to other neurons, but in connections to muscles. Those are the axon terminals there. So here we have the basic structure of what was called at that time the reflex arc. And in many paradigms, this has been understood as sensory input. The interneuron is where the brain is, where consciousness is produced, and the motor neuron then causes the muscles to dance. That primitive picture is no longer a good characterization of the nervous system. Nevertheless, that's the picture that's emerging here, and these long tendrils that neurons have, these axons and dendrites, are extremely important here. Now, this development of Golgi staining and the resolution of individual cells within the nervous system was of such import that the two characters, Camilo Golgi and Santiago Ramon y Cajal, were jointly awarded the Nobel Prize in Physiology or Medicine. This was the first time that prize had been shared, and it was awarded in 1906. And it must have been rather funny, the ceremony, because not only was Ramon y Cajal slightly better draftsperson, the two guys didn't agree on much. They didn't agree on how we should understand the basic modus operandi of the nervous system. So for Golgi, the basic thing to understand was a network of neurons with their intertwined axons. This, he thought, was the level at which we would gain understanding into the role of the nervous system within the overall economy of the body. Below that, there is, of course, detail. There are individual cells, but he thought this was more or less irrelevant, and it was to these networks of neurons that we should look if we want to understand how the nervous system plays its role. Ramon y Cajal, however, thought that the basic functional unit of the nervous system was the individual neuron. Now, this distinction is of theoretical importance, great theoretical importance, and it also lies in the simple empirical distinction between these two guys, where Cajal had made clear that there were small gaps between the dendrites of one neuron and the cell body or axon or wherever it attaches to the next neuron. They stood up to give their speeches at the Nobel Prize ceremony, and they trashed each other. It was very funny. So the debate was unresolved in 1906, but over time, Ramon y Cajal's view won out, absolutely. It became the orthodox way to understand the nervous system, which is that it is composed of neurons, each neuron being in some sense independent of all other neurons. That has become known as the neuron doctrine, and we know it to be a radical simplification. We have great reason to be cautious about adopting this simplistic view, but it was a very important and influential first start. So, your basic neuron then receives input, assuming it's not a neuron that is connected to receptors. If it's an interneuron of any sort, it receives inputs through the dendrites of other neurons, on the order of 10,000 other neurons per cell, for example. Those inputs sum. That is, the cell does nothing until the entire input, summed input from all the other neurons reaches a critical level called a threshold. And then when that threshold is passed, an electrical spike is generated, which propagates down through the axon to the downstream neurons, terminating in the dendrites. It's called depolarization. At that point, chemicals are released which stimulate the next neuron down. So we have an electrochemical junction. This spike, this release of energy is called depolarization, and shortly afterwards, the neuron enters what's an unresponsive refractory period, as if it had a little orgasm or something like that. It just is, it's not up for anything for a little while, and then things sum again. Now, the typical firing rate of a neuron, the rate at which can generate a spike and then rest and then sum and then have another spike and so on, is typically about 100 times a second, 100 hertz. You might compare, because I know you're going to draw computer analogies here, you might compare that to your average laptop computer, where a central processing unit processes well more than 1 billion instructions per second. So from that point of view, there's no comparison to be drawn between neurons and computers, and we'll be making quite explicit what we do when we draw comparisons between the nervous system and computers. Now, Ramón y Cajal's guess was a very, very good one. It ignored all those non-neural cells and it suggested a single mode of processing. We have found exceptions to this in the meantime. We have found spikes that propagate backwards, the wrong way round. We have found direct electrical connections between neurons without the synaptic gap. We have become uncomfortably aware in the last sort of 15 or 20 years that other cells within the nervous system, the glial cells, are also crucially involved in modulating the activity of the entire system and therefore in information processing. The synapses were characterized in the neuron doctrine as simply a point at which a signal is passed from one to the other. This turns out to be very, very oversimplified. They're vastly more complex than that. And we now know that some groups of neurons do act as functional units, always working together along the lines that Golgi suggested. So the neuron doctrine is not a simple truth. It was a very, very good first guess, though. The synapse is really important here. This is the point at which a junction exists from one neuron to the next. They don't usually quite touch. And the transmission of activity from one neuron to the next is effected through the release of what are called neurotransmitters. These are chemicals released by one and taken up by the other. And the economy of neurotransmitters within a synapse turns out to be very, very complicated. The synapse is really important because the character of the synapse determines the effectiveness of one neuron in stimulating another neuron. And synapses are plastic. They change over time. Because they change over time, these are the principal way in which the nervous system is modulated by experience. And so these have become the site at which we usually think of learning happening within a nervous system. Now, we have to be very careful when we use the word learning. Learning here does not mean that which goes on in a classroom where there's a teacher. A nerve is modified by the activity of other nerves around it. Ultimately, that activity is related to the activity of the organism in its environment. But from the point of view of the nerve, it knows nothing about environments, it knows nothing about the world. It is simply any modification of a synapse happens based solely on locally available information. So learning means modulation of the synapse as it is effective in stimulating the next neuron as a result of what has happened recently. That's what we mean when we say that learning uses locally available information. So here you can see neuron A generates an action potential, a spike, which goes down. There's a small synapse in the box there. And it's one of perhaps 10,000 other neurons which are also connecting to neuron B. And the ability of neuron A to affect neuron B depends only on their connection. It depends only on what goes on inside that box and not on something that happens at a remote site. This particular character of the synapse will be represented in the networks that we will study as a weight connecting two units. So a unit will stand for what we see here in a neuron, and the connection between them will be represented by a single number called a weight. This is a gross simplification, and we will have great reason to refuse the idea that neural networks are, in any realistic sense, models of brains. I mentioned that neurons are not the only player. Here you see an illustration from a fairly recent paper illustrating something called a tripartite synapse. So the two blue elements there are neurons releasing chemical transmitters. The green is an astrocyte, which is part of the glial system. And until about 2000, it was generally assumed that this was more or less just like insulation. In the wiring of your house, electrical wires have insulating layers around them so that they don't pass electrical signals from one to the other and short circuit the whole thing. It was assumed that this was the only role for these glial cells and astrocytes, but it turns out that's not true. A single synapse turns out to be a very complicated feedback-driven system. And finally, just to remember that the brain is not only an electrical signaling system. We will be developing models that are inspired by ideas about brains, in which all of this nuance will get lost. But when we look at a real brain, its modus operandi is electrochemical, and the chemical bit is just as important as the electrical bit, and it's not separable from that. So that all this complexity will find correspondence in our models through this single number of the weight. That will have consequences. The required reading then for this little section is an overview article here of the neuron doctrine from a 2005 standpoint. We have learned a lot, so almost 100 years after the awarding of the Nobel Prize, and you'll see that we've learned an awful lot in the meantime.